you for the opportunity to present today. For anyone who's worked on KATP channels previously, you may be looking at that title thinking, I thought it was 6.2 expressed in ventricular cardiomyocytes. You're right, they are. But we believe that we have found 6.1 channels in those cells as well. And we think they have a role in cardiotoxicity and cardioprotection. And that's what I want to talk about today. So first, a brief overview. These channels are sensors of cellular metabolism. They couple a cell's electrical excitability with the energy availability within the cell. And for this reason, in the heart, they are generally considered cardioprotective against ischemia because a larger potassium influx, which they can provide, can reduce calcium load. There are two main isoforms, 6.2 and 6.1. And the mRNA and protein of both have been shown to be expressed in ventricular cardiomyocytes. Now, the role of 6.2 is well established. We know it's blocked by intracellular levels of ATP, and when those levels drop, for example in ischemia, that channel will open. But on the other hand, 6.1 has been relatively overlooked. We don't know whether it's active or what its physiological role is. So today's talk is split into two main parts. One, to show the evidence that these channels, 6.1, are actually expressed in ventricular cardiomyocytes, and I'm going to show the evidence why we think they have a role in cardiotoxicity and cardioprotection against ischemia. So just to outline the methods, most of the data I'm going to show is from male Wistar ventricular cardiomyocytes or their whole hearts, and two main electrophysiology methods have been used. One, cell-attached patch clamps, where the patches are held at an equivalent potential of minus 110 millivolts, and these are a lot of wholesale current and voltage clamp recordings to measure action potentials and currents. So the first slide that I want to show is just basically outline how this work sort of began and why did we think 6.1 was there in the first place. And so what I'm showing here is a cell-attached recording, and you can see at the end of near the ends of the trace I've added simulated ischemia. This solution just contains no glucose, added acetic acid and cyanide to deplete ATP. And as you can see, you get a 10 picoamp current. That's classic 6.2. It's metabolically sensitive. ATP is fallen. That channel opens. But just before you go into poison, there's that 5 picoamp current. And it was literally just because of that size difference, it was half the size, it was initially suspected that could be 6.1. And what we've been able to show is that 6.1 block reduces the leak current. So in wholesale voltage clamp recordings, if you're measuring the leak current, HMR, which is a 6.2 blocker, have no effect on the leak current, indicating 6.2 channels was shut. But the PNU, the selective 6.1 blocker, did indicate those channels were open. And for further evidence to know that it was 6.1 channels, we've compared the conductance of monomers of 6.1 or 6.2 in HEC cells to our cardiomyocyte analogues. And you can see there's a good correlation between the 6.1 monomers and that 5 picoamp current we were seeing in the cardiomyocytes. And the same for 6.2, with the metabolically sensitive analog. On the right, I'm just showing the sensitivity of ATP for between 6.2 monomers in those inside our patches and dimers of 6.1 and 6.2. And basically, those dimers actually retain the sensitivity of 6.2. So because we've seen 6.1 channels in the cardiomyocyte in physiological conditions, we believe we're not seeing dimers. It is likely to be 6.1 monomers. We've also looked at the pharmacology. So first, I'm showing, again, cell-attached patch clamp recordings in control conditions or with 10 micromolar gabenclamide. And you can see in a concentration-dependent manner, the open probability of that channel is reduced. And the IC50 between the 47 between the myocyte recordings and those on CHO cells expressing 6.1 and the SUR2B subunit was compatible. Similar data was obtained using PNU, the selective 6.1 blocker. Again, in a concentration-dependent manner, you get a reduction in the open probability of that channel. And the IC50 is again compatible between the myocyte recordings and those on CHO cells expressing 6.1 and SUR2B. We've also looked at the activator, pinacetil, and 150 micromolar pinacetil is quite a high concentration, and you can see in the cell-attached configuration, I was only able to detect a change in the open probability of the 6.1 channel, but not on 6.2, even at that high concentration, indicating that there is some sort of window of selectivity for between the channels using that drug. And to be absolutely sure that we were looking at 6.1 channels, we've done some manipulation of the gene that encodes those channels. First, we used an shRNA adenovirus, and you can see, again, I'm showing cell-attached recordings, either in control virus, which had the scrambled sequence, or with a knockdown virus. And you can see the open probability is reduced, indicating that these were 6.1 channels. And to be absolutely sure it was 6.1, we've then used the knockout mice from Andrew Tinker's lab in London, and it was only in the wild-type mice that we were able to see 6.1 openings. So 6.1 is a weak inward rectifier, so we hypothesised that as it's active, it could be modulating the cardiac action potential and calcium transients. And using current clamp recordings to measure the action potentials, we were able to show that you get a prolongation of either the 6.1 selective blocker PNU or gabanclamide. And using flow 4 to measure calcium transients, we were able to show a change in the calcium transients in terms of amplitude and area under the curve, indicating more calcium was coming into the cell, likely due to that prolongation of the action potential. Conversely, if you potentiate 6.1, you can reduce or shorten the action potential duration, even with concentrations as low as 30 micromolar per nastal, much a significantly lower concentration that you need to activate 6.2. And with concentrations as low as 50 micromolar, you're able to detect changes in terms of amplitude and area under the curve with the calcium transients. Interestingly, with 200 micromolar per nastal, a few of the cells actually didn't show calcium transients. They actually failed. That's likely because the action potentials failed because of 6.2 activation can actually cause a failure of the action potential. And so just to summarise so far, we believe that these two KATP channels, you've got 6.1, it's open at rest, it can be blocked by drugs such as PNU and gabanclamide, and relatively lower concentrations of pinacidil can activate that channel as well. High concentrations of pinacidil will obviously no doubt activate 6.2 as well. And in metabolic stress conditions where ATP is depleted, 6.2 will certainly activate. And so at that point, we hypothesised that in generally, as these channels are considered cardioprotective, does 6.1 block actually induce cardiotoxicity? And to investigate that, we've used a phenotypic screen of simulated ischemia. Just to walk you through this protocol, at the start of the experiment, we've got rod-shaped cardiomyocytes. We apply electrical field stimulation at 1 hertz throughout the protocol, and we can measure contraction. We then go into that simulated ischemia phase, no glucose, cyanide, and hydroacetic acid, so ATP will be depleted using that solution. And by the end of that protocol, they've gone into this Rigord shape, they're calcium loaded, and they can't pump that calcium out because the ATP is depleted. Once we go into that reperfusion phase, these two options, if the cell is so calcium loaded, it can actually go into hypercontracture. The contraction is so great, as it starts to produce ATP again, it can actually cause cell damage and cell death. Or the cell can recover its calcium homeostasis and regain its contractile function. And at the very end, we can use trypan blue to measure cell death and cell survival. And what we've been able to show is 6.1 block is cardiotoxic in these simulated ischemia experiments. At the start of the experiment, you've got your cells contracting nicely to the electrical field stimulation. In the simulated ischemia, they will fail to contract, and a proportion of cells will recover in the reperfusion phase. But the proportion that recover their contractile recovery is reduced in the presence of either gabanclamide or the selective blocker 6.1, and similar data for cell survival, it's reduced in the presence of gabanclamide and PNU. We backed that data up with a whole heart model of the left anterior descending coronary artery ligation. Using this model, what we do is we just allow the heart to normalise for an hour first, followed by a 40-minute ligation of the artery, and then there's a 3-hour reperfusion phase, and then at the very end we retie the suture and apply the Evans blue dye. And once we slice the heart up, we can see 3 areas. One, we can see the blue area that was not affected by the ligation that we applied. We're not interested in that area. And then we've got the area at risk that was supplied by the artery, we ligated, and then you've got this necrotic area, this infarcted area that was damaged because of this process. And the presence of PNU increased the infarct size, indicating 6.1 block was cardiotoxic to the whole heart. And we think this block could be clinically significant because of drugs such as like rosiglitazone, which was withdrawn for clinical use. And it was withdrawn because of increased likelihood of death from ischemic insults in some circumstances. A few mechanisms have been proposed, but we believe 6.1 could be one of those mechanisms. What we've been able to show is that rosiglitazone blocks cardiac 6.1 channels. Again, I'm showing cell-attached recordings in either control conditions or with 30 micromolar rosiglitazone. And you can see in a concentration-dependent manner, rosiglitazone decreased the open probability of the channel. We supported this by showing rosiglitazone inhibited the pinacetyl-activated current in CHO cells. And in the simulated ischemic assay, rosiglitazone worsened the outcome in terms of contractile recovery and cell survival. What's interesting is the related compound pioglitazone had no effect on the open probability of the channel and had no effect on the simulated ischemia assay, indicating that rosiglitazone block of 6.1 was cardiotoxic. And so, if block of the channel was cardiotoxic, then perhaps potentiation of the channel could be cardioprotective. And what we've been able to show is low concentrations of pinacetyl are cardioprotective. Either using 50, 100, or 200 micromolar of pinacetyl, you can see an increase in contractile recovery and cell survival. And what's interesting, I think, is with the time to contractile failure, the time is it's much less in the 200 micromolar pinacetyl. We believe that's because you get an activation of 6.2 as well as 6.1, and 6.2 can cause action potential failure. So we think that the presence of that drug at that concentration with the simulated ischemia causing ATP depletion actually causes the failure to happen earlier compared to the 50 micromolar, whereas you just, what 50 micromolar pinacetyl is doing is perhaps just potentiating the current to just reduce calcium load, but having no effect on 6.2. And what we've been able to back it up with the whole heart experiments as well. Again, the coronary ligation, and you can see 10 micromolar pinacetyl actually decreased the infarct size, indicating that potentiation was cardioprotective. However, I must say that 6.1 is expressed in the vasculature, so it could be attributed to that, to the potentiation of the vascular isoform. So because we've shown potentiation, direct potentiation was cardioprotective, we then thought that perhaps classic cardioprotective stimuli could lead to an increase in 6.1. And these two methods we've used to cause cardioprotection, one is ischemic preconditioning. That's just 3 short rounds of ischemia will actually cause a protective effect to a prolonged period of ischemia later on. And the other way we've cardioprotected the cells is just with a 5-minute pre-treatment of adenosine. On the right-hand side, I'm just outlining some just key properties of cardioprotected cells. The time for action potential failure, calcium transient failure, and contractile failure are all delayed in the simulated ischemia because they are cardioprotective. Interestingly, the opening of 6.2 is also delayed, and that's likely because KATP levels are preserved for longer, and so that channel remains shut for longer in those cardioprotected cells. And what we've been able to show is IPC cardioprotection actually increases the open probability of the channel. If I could just compare cell-attached recordings in the control conditions to the IPC cells, you can see there's an increase in the open probability of the channel, and that increase was, again, sensitive to both PNU and glibenclamide. And as you expect, if you potentiate a weak image rectifier, you actually get a shortening of the action potential duration, which is what we've observed between the control cardiomyocytes and the ischemic preconditioned cells. And the KATP blockers actually attenuate IPC-induced protection. These are, again, the simulated ischemic assay. The cells are contracting at the start. They will fail during simulated ischemia, and a proportion will recover in reperfusion. But the presence of PNU or glibenclamide reduced contractile recovery and cell survival, indicating that 6-point block attenuated the the protection afforded by the IPC protocol. And again, we've backed that up with the whole heart method, where even the IPC hearts, if they were in the presence of PNU, had an increased infarct size, indicating that 6.1 block attenuated the protection. We also got similar data with adenosine pretreatment cells. Again, you get an increase in the open probability of the channel. You get a shortening of the action potential duration, and PNU attenuated the adenosine-induced protection in the simulated ischemic assay. Gone quicker than I thought here. You always go quicker. So this is my concluding slide. So none of the data that I'm showing is to suggest that 6.2 in itself is not cardioprotective. What we think is actually that there's two cardioprotective KATP channels, just by slightly different mechanisms. So what we think is going on is you've got 6.1, which is open at rest. It can be blocked by drugs such as PNU, glibenclamide, perhaps rosiglitazone, and that can be cardiotoxic. On the other hand, if you potentiate the channel either directly with smaller concentrations of panacetyl or indirectly, as we've shown with IPC or adenosine, that potentiation can actually cause a shortening of the action potential duration, which leads to reduced calcium influx. And that improved calcium handling could be important in ischemic conditions where ATP levels are critical. If calcium handling is improved, less ATP is then needed to pump out that calcium. And as ATP is preserved for longer, 6.2 channels remain shut for longer. However, if that ischemia persists, 6.2 will open. And what we think 6.2 is doing is it is the last-ditch attempt of the cell to survive. It will cause a very large hyperpolarization to the point you get action potential failure of the calcium transients, and contractile failure. And it's literally just a last-ditch attempt of the cell to try and survive and prevent calcium overload. I'm not going to name everyone, just because there's too many, but thank you to everyone who's contributed to this work. This has all been done in the lab of Dr. Richard Rainbow, and I'd like to acknowledge the British Heart Foundation for funding this research. Thank you very much.
Okay, thank you very much, Sean, for a lovely talk and fantastic recordings. Was that you doing those cell-attached recordings?
Yes.
Fantastic. Brilliant. Do we have any questions? Yes. Wait a second, please, for the microphone.
I was just wondering, in the absence of an activator or cardioprotective stimuli, what actually causes KIR6.1 to remain activated in ventricular cardiomyocytes?
So, you mean what causes the increase between okay, so we're not sure what causes the increase in between when we cardioprotect the cells, but just to show you this data, whatever the change is, it does happen very quickly. So on the right-hand side, I'm showing that if I just do cell attached recordings again, I can then just wait till I activate 6.2, so I know I've metabolically stressed the cell to some level. If I quickly then go back to normal thyroid solution, I can see just even an increase in 6.1 activity there. And I know Andrew Tinker's lab in London has shown that the 6.1 channel can be metabolically sensitive as well. So it could just be simple, just subtle changes in metabolism that the cell, that the channel is detecting. It could also be in the vasculature, for example, protein kinase A and protein kinase C can modulate this channel, so it could be something like that. And we've shown that actually 6.2, I showed the 6.2 delay was actually sensitive to PKC epsilon TAT peptide inhibitor. So it could be PKC epsilon, which is known to be involved in cardioprotection as well. So we're not sure on the mechanism, but these are few potential ways that it could be increased in cardioprotected cells.
That's great, thank you very much.
Cheers.
And isn't the 6.1 got a lower sensitivity to nucleotide diphosphates? So NDPs like ADP, UDP. So there's going to be pretty decent resting level of that.
Yeah.
If I remember rightly, I think 6.1's got a lower sensitivity or better sensitivity to the diphosphates than 6.2. So that might allow you to have that channel at rest.
Yeah, and then it could change. It's just those subtle changes, perhaps.
With the
Yeah.
mechanism itself. Okay. Any other questions? Yes.
Yeah, I've got the microphone.
Oh, thank you.
Can you hear me okay?
Yeah.
I just wondered from the modelling point of view, I think this is quite challenging. How did you decide what concentrations of rosiglitazone were relevant to test in vitro? How do they compare to the human subjects who suffered, cardiotoxicity?
So I think it's about 2 micromolar clinically. So it is a bit higher, but I think rosiglitazone can be used with glibenclamide clinically as well, and I think that's when it became clinically relevant in terms of the concentration. So I think it is a little bit high, but the block was there. So it is just one potential mechanism of a few that have been suggested for the reason why rosiglitazone caused that increase.
You're looking at acute effects, yeah? You're just
Yes.
hours in vitro versus long-term use?
Yes, absolutely, yeah. It's just literally applied before, during the experiment.
Thanks.
Okay, so thank you very much. If you have any questions, please see Sean afterwards at lunch, wherever. Thank you very much, Sean.
Thank you.